Thanks for having me come back for some updates. It's been a while since I last shared information with this group. We talked in October. I gave you an overview of all of the NIH guidelines, which really focused on therapeutics. And I'll give you a quick update of what's changed since then, because this does, of course, change rapidly. And then I'll jump into what I prepared is just the most common questions we seem to get about vaccines. What I'm showing you right now is something you might be familiar with from the literature, and that's a schematic of the different phases of COVID-19 disease and how we try to fit different therapeutic classes into that. And so, early after infection, initial infection with the virus, replication starts happening. You have this, we're starting to call it a viral response phase. Right there, right away, is the sweet spot for antibody therapy. Before someone's been exposed, before someone has a reaction and the virus really takes hold, there's an opportunity to use antibodies directed towards the virus to try to prevent progression. So, the therapeutic agents there are still evolving, and I'll highlight a couple of those to you. That's also where we might be able to use antiviral therapies. But as anyone who works in the antiviral space knows, we don't do very well with antiviral drug development. These are a lot more challenging than even antibacterials to really narrow down and figure out what's going on. So this is still not a lot of antiviral options. As that virus starts to dig its feet in, and patients start to have a reaction, that pulmonary infection and inflammatory phase, it was what's causing long-term poor prognosis. So about 10% of patients do progress to requiring hospitalization, oxygen, and even mechanical ventilation, very critical illness. Those who recover are having prolonged and profound effects of this disease. Even when they recover, we are seeing this syndrome called long COVID, characterized by pulmonary fibrosis. There's a lot of need for therapy for these patients long after even recovering from the acute illness. So we've got a lot of questions on both ends of what do we do early, what do we do in critically ill, and we don't know anything much about what we do later. So those are areas that I see people from this college still having a tremendous impact on, with the science and practice. Then, of course, before all of this, before that infection, that's where we want to target people with vaccines. So the last time we talked in October, I showed you the initial draft of the NIH guidelines, where there are treatment recommendations for each phase of disease severity. These haven't changed much, but I'm going to point a couple of things out that we've seen some movement on. And the first is at the severe end of the spectrum. So we're narrowing down what is most beneficial in severe disease, finding in more updated studies that remdesivir might not be as beneficial as initially guessed. That's the antiviral drug that we use in hospitals for patients who are hospitalized. But once this disease progresses to critical illness, severe illness with inflammation, it might be a little bit too late for that to be useful. So remdesivir is not being recommended anymore and those who are very severely ill, critically ill with mechanical ventilation. But that leaves questions about what do we do for these patients? We're using steroids. That's still having a mortality benefit. That's still the mainstay. But what do you do if patients don't respond well to steroids, or if they can't take steroids? And can we do better? And so that's where the question of some of these more selective immune modulatory therapies works. Steroids are pretty broad spectrum in their anti inflammatory response, but there are quite a few other classes that target specific mechanisms. And so some of those that are getting out into guidelines and use right now are baricitinib, has an emergency use authorization. It's mostly being used as an alternative to people who cannot tolerate steroids. And then in the past few weeks, we've seen new data coming out on these interleukin 6 inhibitors that are used in rheumatic diseases that might provide an additional benefit on top of steroids, either when they're used early, then some questions about steroid refractory disease. That's where practice is changing right now, right this week across the country. People are updating their guidelines in response to those studies. So that's exciting to see some new data for those who are most critically ill. But the biggest gap that I think we have is what to do for people with mild to moderate illness. Patients who are in the outpatient setting, they're not sick enough to go to the hospital. We kind of have nothing. Like, there's not much that we can say this helps these people reduce their duration of illness. It's like a really bad cold or flu. So, symptomatic relief, the things that you would recommend to patients in outpatient pharmacies and primary care settings, the same symptomatic relief is basically all we're doing. But is there anything that we can do to prevent progression to hospitalization? That's a big question. So the first line of this slide are representing some monoclonal antibody drugs. These are antibodies specifically directed at the virus, bamlanivimab and casirivimab imdevimab. So these two products have been authorized for use in outpatients who are at high risk of progression. The data is not final on what things are going to look like, but people across the state of Michigan, particularly northern Michigan, have been having success in taking patients who are at high risk outpatients, using these monoclonal antibodies and reducing the burden of overall disease. The other options here are things that people are talking about out in the world that don't really have a lot of data. So the first, ivermectin, is an antiparasitic drug used for strongyloides in a number of countries. It's proposed to have antiviral and anti-inflammatory effects. And the pharmaceutical scientists working on this can show that in vitro doesn't translate quite so well to clinical data. And so the current statement that we have at the NIH guidelines is there's really insufficient data to draw conclusions. And if you are looking for a place to go critique literature, this is a great place for it, because the publications and preprints are a mess. There's just giant gaps in their studies. The other two drugs that are coming up more commonly in conversation now are fluvoxamine, an SSRI, and colchicine, which is used for gout, pericarditis. Both of these potentially have anti-inflammatory effects and are being evaluated in small studies and gaining a little traction as could these prevent progression. No answer on that now, but this is where a lot of focus on new studies is. What can we do for those majority of people who are not that sick, but we don't want them to get sicker? So that's my therapeutic updates. But the hot topic, the fun topic that I think everyone is really focused on, are the vaccines. So I'm going to start briefly. Yeah, I just came up with some questions that come up a lot, and there are definitely people on this call who know at least as much, if not more, about these things. Happy to hear from Paul, Brittany, Mike, all of the other folks. Um, how do these vaccines work? There are four primary ways that different vaccine platforms are brought to market. And the central concept in all of them is that what you want to do is expose the body to a harmless part of the virus that will induce an immune reaction and prevent future illness. One of the big questions in patients and or the community is this mRNA mechanism of the vaccines that we're seeing in the market right now. And I think in part of that is it kind of sounds like science fiction that you're using mRNA, but it's not science fiction. It is science. So once the vaccine is injected, mRNA is taken up by white blood cells around the injection site, and the mRNA is like a message that tells the cell to make the spike protein that you see on the outer blob of the virus particle. That spike protein then appears on the surface of those white blood cells, where it's not going to do any harm, but it does induce an immune response. Then that mRNA that was in the vaccine goes away. Enzymes in the body naturally degrade it and dispose of it. No live virus ever enters the body from these vaccines. No genetic material enters the cell nucleus. It would be pretty impossible for these mRNA vaccines to somehow alter cellular DNA. And that's one of the rumors that I think people are very unsettled about, is how do you communicate that mRNA does not alter your genetic material? So I've had some people describe it as like a Snapchat message that you see it and it goes away. I don't relate to Snapchat. I think of it like a post-it note. So, I leave myself a post-it note of all the things I'm going to do today. I do them, and then I toss the post-it note. That's what these mRNA messages are. It's a post-it note. The cell does the thing and then tosses the message away. So that's how I tend to understand it, or people of my generation, I think, maybe would understand it. I think the next big question, if that's how they work, how well do they work? And communicating this, I think, is really important, that we're communicating the degree of protection and what it's protecting against. So the Pfizer and Moderna vaccines have 95-ish% protection, 95-ish% efficacy in preventing disease. If you look at the curves that come from those studies, that curve of going up in each of those is the unvaccinated population progressing to symptomatic disease. These do not reflect a difference in transmission. It might protect against transmission, but that data has not been fully vetted yet. So we cannot say that these vaccines prevent transmission. But in terms of preventing disease and the subsequent death, they're highly, highly effective. The Oxford vaccine from AstraZeneca is running around 70%, and the other vaccine platforms are still really under evaluation from the companies in the FDA, and so that information isn't public yet. But if you're looking for a comparison of how does this stack up against other vaccines that we know, the flu vaccine that we get every year is usually 40 to 60% effective, depending on the year and the strains, and that still is a major public health benefit for us. This is much more analogous to the MMR vaccine that we would get for preventing measles, or even the polio vaccine. Those were at the 97 to 99% effectiveness. So these Pfizer and Moderna vaccines are very high on the scale of effectiveness in preventing disease, which is pretty exciting. Another question, you see stories in the media, and if you're reading the literature, you hear about these new strains from South Africa, from the UK, from South Korea. There are new strains, new mutations, some of them of serious epidemiologic concern. So will the vaccine protect us against those? I don't have a great answer for you. What I can say is that these trials were done before we knew about these mutations and these new strains, so we can't say with the same degree of certainty. There are a few studies that have been done. There are preprints shared on the immunogenicity with these, suggest that our current vaccines should be somewhat protective. It will vary between the strains. And so keep paying attention to this. The companies who are developing the vaccines are currently working on adapting their formulations, which is great for advancing our technology. And a possible booster dose might be something that could help enhance the immunologic impact here. Another one of the questions that I'm hearing among our students and my colleagues when we're offered a vaccine, some people are saying, I want to wait because the studies were rushed. I want to wait because the studies might have been influenced by political interests. And I think when people are expressing this type of hesitancy, it's bringing up a couple different questions. And one is, we don't necessarily know everything there is to know about the short-term and long-term safety and efficacy. And that's quite true. We don't know what the long-term effects will be. However, in the risk benefit of vaccine information and COVID outcomes, to me, there's just no comparison in the risk to benefit. And so when that's the reason people are questioning things, I do try to talk about, if you had COVID, things are going to be pretty bad in the outcomes. This also brings up issues about trust in the pharmaceutical industry and government and regulatory agencies. For us in our community, look around the college. You've got people who are doing these studies. You've got people who are directly involved in the care of these patients, in the rollout of these studies. And I don't know about you, but I look at my partners in this building, and I see a lot of ethical people that would not be letting something sneak by. So I feel comfort in that personally, knowing people working in this area. I also would point out a little bit about what exactly is rushed. So I didn't feel like creating these graphs again myself. So I stole from a colleague who created some presentations for SIDP. The normal vaccine timeline is taking several years of preclinical development, and then the clinical trials do typically take a long time. Whereas a previous 5-year study time frame has only taken about a year or less in our scenario now with these COVID vaccines. But that's not a difference in the man hours. I think if you talk to people who've been involved in vaccine clinical trials, the number of man hours and the patients enrolled and the observation periods in that 5 to 7 years really isn't all that different from what we're doing with these studies now. The degree of intense scrutiny is still there. It's not as much time on the calendar, but it's as much time in man hours of people who are looking at this. If you looked at my calendar, I'm not doing vaccine studies, but I would not normally spend 200 hours reading about ivermectin in a month, but that's what I did in the past 2 months. Normally, I would spend maybe nothing. I would spend none of that time. So I think that shift in focus has really allowed us to do some really good work in a very short calendar time. I do have confidence that people are doing the right things with these study designs. The issues about shortage and a second dose and what to do if a second dose is not available, that is something to be worried about. So the CDC has been evaluating some of these things, and the first dose does provide some protection. Delaying the second dose might still be just fine if we are forced into that situation. Likewise, if we run out of Pfizer but have Moderna, they have now reversed their initial position to say, it might be okay to switch formulations. So doing what we can with the products we have. I am very excited to see the Defense Production Act ramp up so that hopefully we can get more product out. So that shortage thing is a great question because it brings me to one of my next questions that I think is fun, and what's up with the extra doses. So if you've ever worked in an inpatient pharmacy as a technician or something, you're familiar with the concept of overfill. The vials that we get usually have a little bit extra in there. It's really good about drawing out the contents. You can get a little bit more of the volume out. The other thing is, if we have the right type of syringes, that helps. There's these things called low dead space syringes. And so I put in an image that some great engineering of trying to minimize the dead space in the syringe can help us sneak out every drop of vaccine in those products. So that's where those extra doses are coming from. Um, there's a lot of questions about who can and can't receive the vaccine, that you can't get it if you have allergies, that you can't get it if you're pregnant, that you can't take it if you're on a blood thinner. The allergies are a real important question. There are some immediate allergic reactions that people have to vaccines. And so the contraindications would be if you've previously had a severe or immediate type hypersensitivity to a previous dose of a COVID vaccine, or to components related to polyethylene glycol. And a lot of people don't know that. If you have a history of anaphylaxis to another vaccine, that's not a severe contraindication, that's not a direct contraindication. Those patients should be monitored longer. And so, for those who are working out in the community in vaccine clinics, that's really the thing that happens. Pregnancy is not a contraindication. Many pregnant women are obtaining the vaccine. It is recommended by those organizations. And blood thinners is a hard thing. Those patients don't have necessarily any additional risk of adverse effects, but because of potential bleeding and bruising at injection sites, they were excluded from some of these studies. That is not a direct contraindication, something just to be more aware of the type of reaction you can get at the injection site. But patients on blood thinners can just need a little bit of additional monitoring and counseling. Some other questions about who can and should receive the vaccine. What about people who've already had COVID 19? The recommendation is if a patient has recovered from COVID 19, they absolutely can and should still be eligible for the vaccine, but the timing is a little different. Reinfection is uncommon within 90 days of the initial infection. If they've been treated with antibodies, that can definitely limit the effectiveness. So, waiting until 90 days after the initial infection is the general recommendation vaccination for people who've already had COVID. The other big population of question is immunocompromised patients. And because immunocompromised patients are at significant increased risk of COVID and of bad outcome if they have COVID, CDC and cancer societies actually recommend vaccination unless there's a contraindication. The big question here is not should they get it, but how effective will it be? Because the immune reaction depends on having a functioning immune system. So that's where the study is needed and ongoing. You can get more information from that from the American Society for Hematology. They've got a great website. And other questions are about how long does immunity last? When does protection start? From my understanding and my reading, the mRNA vaccines, we start to see some immunity after 10 to 14 days after the initial dose. We would consider the more full, comprehensive immunity at least a week after the second dose. Now, how long it lasts is something that evolves with time and monitoring. So, based on the Moderna study, we would say most patients had um, eh, prolonged protection for at least 4 months. Some follow up data suggests probably 8 months. And as we continue in time, it might be even longer. When new products come to the market, though, we might find that this varies between products and varies between people. Right now, there's no recommendation for a booster. That might happen in the future. And right now, there's no recommendation for a seasonal vaccine, but that's entirely possible that this might be something that we need periodic updates if COVID stays in our communities, which I would love for it to go away and never to have to talk about this again. Leave these questions unanswered and let's all be fine. But let's be realistic. This isn't going away soon. One of the questions patients often have is, can we go back to normal and stop wearing masks after I get a vaccine? And as you might have seen from some of the media reports out here, not yet. And so there's a couple reasons for that. The outcome these prevent is getting sick, not acquiring the virus or potentially transmitting it. So we don't know how well the vaccines will protect against transmission. The other thing is we don't have nearly the vaccination of our communities to protect others. So masks are still essential in those group activities, masks and social distancing, even as we vaccinate a larger proportion of our population. The last slide I have for you is the misinformation that you see out there. So I've heard the vaccine has a microchip. I don't know where that came from. I really, that's like from a Facebook post. Um, it doesn't have a microchip. That doesn't even make sense to me. Um, but this is a real fear that people in my family have, people in our community might have. So when you hear that, my initial reaction is to cringe, but I think we have to listen and try to hear where it came from, go back to the source and express both empathy and then confidence in what we know about the vaccine, that there is not a microchip. There's the rumor that it's caused a bunch of deaths, and I know where that came from. So there are reports, first in Norway, the Pfizer vaccine, after over 40,000 people had received that vaccine, they reported a cluster of 33 deaths in nursing home patients. Norway prioritized nursing home patients first. And so I think one of the things that you see there is that these were elderly patients, many terminally ill, and that early death after the vaccine was ruled to be likely natural causes. The same thing was reported in Germany. There were 7 older adults who died shortly after vaccination, after 800,000 people were vaccinated. So the World Health Organization took this to heart and did a deep dive into the epidemiology here. They found that the all-cause mortality rate in those patients with their underlying conditions and age was no different than what would be expected of the normal population. And on top of that, for those of us who do care for elderly populations, on balance, the risk benefit is still favorable in terms of getting the vaccine to those people. Another rumor is that it causes infertility. And it took me a while to figure out where that came from. But there's a German epidemiologist who has a history of vaccine skepticism, which, okay. And he petitioned the European Medicines Agency to delay approval of the Pfizer vaccine because there's a protein in the Pfizer vaccine that is also in the placenta, or at least shares some structural components. Their argument is that if we have antibodies to the vaccine, or for the vaccine, so antibodies to that protein, would we not have antibodies to a placenta and cause infertility? There's not a lot, that doesn't make sense for people who actually do this work, but anti-vaccination blogs and Facebook really let that take off. So because of the worry about that, the CDC and other agencies are doing enhanced monitoring of fertility and pregnancy in those who are vaccinated, but there's zero evidence to actually support that this would happen. The other thing that I think is interesting is Amazon Prime had a TV show, Utopia, that at the same time this is happening in our real world, this TV show had a vaccine storyline of using vaccines for population control. Very sinister and dystopian. So all of these questions swirl throughout communities, and I think data doesn't necessarily change minds, but stories do, empathy does, listening does, letting people vent. And what my process is with family and friends is to use the SIFT approach. I don't know if you've seen this for misinformation. So stop cycling, stop retweeting, look at the source. So investigate the source. Where did this come from? Why are we talking about this? Find better sources, better informed sources, actual data, and trace those misinformation to their sources to come back to what is real, and stopping that cycle of misinformation. I definitely want to highlight Brittany and Paul and everybody else who's doing work with these studies. Mike, who is just such a great communicator with the community and with people about infectious disease. I think we've got some really great expertise.
Susan, thank you so much for all of the science, the clarity, and your communication powers. They're excellent. I think I saw something from Joe saying he's going to use your sticky note analogy. I'm going to too. It's wonderful. It is of great interest to all of us. We should all be knowing this as deeply as we can with our students too. Very much appreciate your contributions on behalf of everyone on the call.